Good afternoon, everyone, and thank you all for joining us this afternoon. And welcome to the 20th Annual Astute Clinician Lecture. My name is Paya Sayalao. I am the Acting Chief Executive Officer of the Clinical Center, and I'd like to again welcome you. Today's special lecture is an NIH Director's Wednesday afternoon lecture series event hosted by the Clinical Center, affectionately known as the House of Hope. We are pleased, I'm indeed very pleased, to welcome you in person and all participants who are viewing the lecture via the NIH videocast. We will begin with some general guidance. Today's code for continuing medical education credit is 66831. Please test the code to the Johns Hopkins CME phone number on the slide to receive your CME credit for this lecture. I believe it's out there, 66831, to obtain your CME credit. For those viewing remotely, you may submit your questions to Dr. Bonnemann anytime during the lecture by clicking the Send Live Feedback button located below the videocast window, and Dr. Bonnemann will answer the questions as time permits at the conclusion of the lecture. For those in the audience, we have two mics, one on my left and one on my right in the middle here. Please, by all means, we encourage you to ask the questions. Dr. Bonnemann has got all afternoon to field those questions. They promised me that during the luncheon, so we're going to hold on to it. But jokes aside, though, seriously, please, for those in the audience here, we got two mics here, and we got Chris here that's going to help facilitate that. The Astute Clinician Lecture is established through a gift from the late Robert W. Miller and his wife, Haruko. I would also like to thank and acknowledge Dr. John Gallen, whose leadership in establishing the lecture with the Millers and in navigating a partnership with the FAES, the Foundation for Advanced Education in the Sciences. We lost Dr. Gallen last year, but his support of this program, of this lecture, and his support for clinical research enterprise, really lives on. His legacy truly lives on. The Astute Clinician Lectureship highlights exciting clinical observations that have served as a focus for study. It honors a U.S. scientist who has observed an unusual, and I will add significant, clinical occurrence, and by investigating it, it has opened an important new avenue of research. It is now my distinct honor and pleasure to introduce this year's speaker, Dr. Kerstin Bonnemann, a senior investigator in the Neurogenetics branch and chief of Neuromuscular and Neurogenetic Disorders of Childhood section of the National Institute of Neurological Disorders and Stroke, NINDS. His presentation, titled Clinical Encounters in Neurogenetics, Taking Cues from Patients to Discover Genes. Number one, to discover mechanisms and therapeutic approaches, would describe three personal journeys of the discovery that are led to Dr. Bonnemann and his team from encountering a single enigmatic patient to uncovering new genes. Not only that, he will also be talking about how these have led to discovering new mechanisms and new treatment approaches, while also providing fundamental insights into human neurobiology. Dr. Bonnemann graduated from medical school at the University of Freiburg in Germany and trained in pediatrics in Hamburg and Göttingen, with a follow-on training in child neurology at the Massachusetts General Hospital and Harvard Medical School. This was followed by a postdoctoral and neuromuscular training at Boston's Children's Hospital. He was the co-director of the Neuromuscular Program at CHOP, Children's Hospital of Philadelphia and University of Pennsylvania Medical School, University of Pennsylvania School of Medicine, before joining the NIH in 2010 as the chief of Neuromuscular and Neurogenetic Disorder section of NINDS. Dr. Carsten Bonnemann research develops gene and RNA targeted therapies for early and onset neuromuscular and neurogenetic disorders. Most of the disorders his team studies are rare or ultra-rare and not typically commercially viable. Clinical genomics and translational work in his section includes first-in-human intrathecal and intravenous AAV-mediated gene transfer trials. His section aims to show, with examples and templates, that precision therapies for a few patients or even one are visible and when the scientific rationale is strong. Dr. Bonnemann has received numerous awards at NIH, including NINDS, NCCIH, and NIH Director's Award. He was a Pew Fellow and received the Denny Brown Neurological Scholar and George Jacobi Awards from the American Neurological Association. In addition to that, he has received the International Cheney Health Prize of the German DGM. And he was also he received the first ever Muscular Dystrophy Association Legacy Award for excellence in clinical research. Dr. Bonnemann has been elected to the American Association of Physicians, AAP. He has served as the communicating editor of the journal Human Mutation from 2020 to 2023. He is currently the co-editor-in-chief of the Journal of Neuromuscular Diseases and has served at several boards, to include the Annals of Neurology, the Journal of Muscle Research and Cell Motility, and last but not the least, the Journal of Neuromuscular Disorders. Ladies and gentlemen, please join me in giving and welcoming Dr. Carsten Bonnemann.
Thank you. Okay. Thank you very much for this kind introduction and this tremendous honor of this award. This is really very close to my heart because I hope you get a sense during this lecture that I am a clinician by heart and remain a clinician by heart, and I think everything we do in human medicine is powered by the encounter with patients. So you'll see my own story with that. I want to start by just highlighting a few kind of personal imprinting events, if you will, that got me into what we are doing right now. So this is Freiburg in Germany, and the Black Forest is there in the background. This is my medical school, when I went there for medical studies. And over here, if you go in that direction, is the medical campus of the medical school. And it's interesting, in German medical school, as a first-year medical student, you have to be a nursing intern for a summer. So you basically get assigned the lowest position on the nursing totem pole in the hospital, and you have to kind of live through that for a summer, which I think is actually an excellent idea because you get to see an aspect of medicine that, as a medical student, aloof as you are, you don't really encounter. And so I got assigned to the Children's Hospital of the University of Freiburg, and not only that, I got randomly assigned to a pediatric neurology ward. And that was my first imprinting event, the first year of medical school. Never looked back. I wanted to become a pediatric neurologist from that moment on, and I've done that. And then the second imprinting event happened after my neurology training at Mass General Hospital. Walter Koroshetz, the NINDS director, actually was my residency director at MGH back at the time. And so at the end of my residency, I knew I wanted to do something with genes. That was kind of as specific as I was. And I walked across the campus at Harvard Medical School and looked at labs that would be doing something with genes. That was kind of the early 90s. And there was Lou Kunkel. Lou Kunkel had discovered the dystrophin gene, solely based on the location in the genome. That was an absolute novum. He really mapped the dystrophin gene, which is a huge, huge protein, like clone by clone, in a handwritten map that's behind him there. There was the first, I think, breakthrough achievement of positional cloning at the time. So I came into his office, naively as I was, and then, would you take me on as a research fellow, and he looked at me, why should I take an MD? What's the point of that? But then he kind of relented and said, well, it's a good idea to have a medical person in a human genetics lab to do that. And he took a chance on me. And it actually worked very nicely. We were able to discover the first limb-girdle muscular dystrophy genes at the time, the sarcoglycans. And this is a time where there was no Human Genome Project, where sequencing was done on hand, kind of pipetted gels, and you had to read the sequence of that. So that was the start I had into not only genetics, but also neuromuscular disease. And for this gene here, beta-sarcoglycan, now 30 years later, there's a gene therapy in clinical trials for it. So that's a nice evolution of what happened. And then the next kind of transformative event was instigated by these two people, that Walter Koroshetz, now Deputy Director at NINDS, and of course Kurt Fischbeck, who had founded the Neurogenetics branch here at NIH. And really what that allowed me to do when they recruited me is to put this in motion, which really was a dream that Kurt had mapped out already, this kind of circle where you start with the patient, the genotype, the phenotype, and you move on to understand the mechanisms and strategies of therapies and the precision therapies as an outcome for that. And this kind of transformative evolution was really powered by the completion of the Human Genome Project and the availability of next-generation sequencing in clinical research. And that really, these two things were made possible here at the campus at NIH. And I'd come from Children's Hospital of Philadelphia, which of course is an excellent place, but this opportunity was really unique, and it led to just a brilliant team that worked with me here at NIH to make this possible. And so what we do here is neurogenetics, and more specifically, neurogenetics of motion and sensation. So this is where this all happens: spinal cord, the motor neuron, muscle, and then the sensory nerve. That's kind of our field, subfield of neurogenetics. And what I want to do in the following lecture is take you a little bit on a journey in that subfield of neurogenetics to show you examples of where an encounter with a single patient really triggers discoveries to gene discovery, mechanism discovery, insights into the human condition, as you will see, but also therapeutic approaches as we go forward. So this is the little diagram here with the motor neuron, the muscle there, and the sensory nerve. So we'll start here with the motor neuron. And this was starting with a young woman who came from Italy to the NIH with an unexplained, very severe, progressive motor decline that she had. And she was sent by her physicians to the NIH to see whether we could figure out what this was all about. She also got the blessing of the Pope. She went to Rome, got blessed before she came, and so she came with the Pope's blessing to NIH to be seen by us. And this is her. So you can see she is a young woman, and her history is that she was 18 years at the time, but she had, from an early age, she had toe walking, so elements of spasticity that were evident when she was a young child. And then following that, she had a fairly rapid decline in motor strengths, leading to a loss of ambulation. At 13 years, you can see respiratory insufficiency underneath this little scarf here is a tracheostomy, a completely normal condition. Cognition. And on examination, what was immediately apparent is that she had signs of denervation. She had fasciculations in her tongue. She had lost reflexes, and she had limb muscle strength that was now no longer able to hold against gravity. And this is not. Hang on. Okay, well, look over here. And on physiological testing, she had on EMG signs of denervation, on muscle ultrasound, also signs of denervation with fasciculations that show up in this video here. if it runs. Yeah. And if you pay close attention to the ultrasound image there, you can see the muscle seems to be moving on its own. It's denervated. It has these fasciculations, clearly pointing to something that involves denervation as part of the weakness. And her sensory nerves are completely normal. So a pure motor neuron syndrome that's evident clinically and on ultrasound and EMG. So we were able at the time to proceed to whole exome sequencing in this nuclear family here of 4. She was the only person affected in this nuclear family. And what we found is that she had a de novo variant in this gene, SPTLC1, that you can see here. It wasn't in her parents, nor in her brother. It was just in her. So de novo variants in genes are always interesting to look at when you have neurologic or genetic syndromes. But the problem was this was actually a known gene. That's a gene that's been already known in the literature as causing the exact opposite of what she had, a hereditary sensory neuropathy. So a pure sensory syndrome without motor involvement. So it was just kind of not making sense that you had a variant in a gene that causes the absolute opposite phenotype of what we saw here. And what really helps, you will be seeing, is genetics. But first of all, let's see what this hereditary sensory and autonomic neuropathy is all about. So this is a syndrome that was described many years ago by a classic neurologist as a pure sensory neuropathy that affects sensory and autonomic functions and had degeneration in the sensory nerves on biopsy. It was almost 20 years ago that geneticists found that this was actually linked to dominant pathogenic mutations in that very same gene, SPTLC1, that we have found in our patients as a de novo variant. And so initially that wasn't kind of making sense, and we were trying to understand why this was there anything else going on? But at the same time, we broadcast our finding across the world to our collaborators and other clinicians who do genetic analysis. And in literally a very little time, it became apparent that this was actually a theme. So here we have 7 families with variants in SPTLC1 and a pure motor neuron phenotype. So that didn't look like a random occurrence anymore. And when we mapped it to the gene and the known mutations for the sensory neuropathy, you can see it maps completely differently. So in red are the mutations for this motor phenotype, this ALS phenotype actually, and then in blue and green are the neuropathy variants. So it clusters in one exon with the same phenotype. That always is suspicious that this is maybe a real connection between the genotype and the clinical phenotype. And this was led by Payam Mohassel in the lab. And highly conserved variants where this happened, missense variants as well as small deletions, but always in frame. And then at the same time, Brian Traynor and Monique Ryan in Australia had also identified single sporadic cases with one of the variants that we also found in the same spot, also with a motor neuron phenotype. So that really thickened the plot in NOMAD in the database for neurological diseases. NOMAD was one patient with a similar variant who actually was listed as having juvenile ALS. So that really meant maybe we are onto something. So amongst these patients that we identified, some in person, some through collaborators, a very consistent phenotype emerged with very progressive, very severe weakness, with the tongue fasciculation, the signs of denervation, the weakness in the limbs, less so in the face, upper motor neuron signs such as clonus and upgoing toes, despite of the weakness, and then also very hypophonic dysarthria, very neurogenic sounding. So that really fulfills classic criteria for ALS. multiple segments of the motor system plus upper motor neuron signs. It's just very, very early in life. Classic ALS is, of course, a much later onset disease, and these are starting to show symptoms typically in early childhood with the toe walking and then this progressive motor neuron involvement. And on biopsy, what you find is signs of clear denervation, grouping of small fibers here, very neurogenic looking atrophic fibers here, but normal sensory nerves. So the biopsy of the sensory nerve, as opposed to HSAN, was normal. Similarly, when you do muscle MRI, what we see here is what we call a salt and pepper or geographic pattern of preserved muscle and fat-replaced muscle, and that is very, very characteristic and typical of denervated muscle. Fits all together as a pure motor neuron sign, a symptom, and syndrome. So I haven't told you what SBTLC1 actually is. SBTLC1 is a subunit of a complex called the SPT complex, serine palmitoyl transferase complex. And that's a very fundamental biochemical lipid biosynthesis pathway that conjugates at the first step an amino acid, serine, with palmitoyl-CoA to form the first building blocks of complex nervous system lipids, including glucocerebrosidase and single myelin that you know from other neurological diseases, such as Gaucher and Niemann-Pick disease, as things that happen when these lipids build up. So this is the pathway, a highly conserved pathway in mammalians, but also down to yeast. These first conjugation steps to the first complex lipid, the ceramide, happen in the ER, and then the more additions to it happen in the Golgi, and then it gets inserted into membranes. So this is a complex that has two major subunits, SBTLC1 and SBTLC2, that sit in the ER membrane with a transmembrane domain, and they have little associated subunits, and we come back to that. And you can see here the structure that's been recently clarified. Come back to that as well. This is just kind of the biochemical steps here in this pathway. And what happens if you use a different amino acid as opposed to serine, another small amino acid that replaces serine, such as alanine, then you develop complex lipids without this hydroxy group because alanine doesn't have it. And that results in species that are deoxy species, so without the hydroxy group. And those are neurotoxic species. And what researchers found early on that this is happening in the hereditary sensory neuropathy. So there the complex shifts substrate from serine to alanine or glycine and makes these neurotoxic deoxy species of it. That was found by several labs and really led to the idea that maybe we can coax the complex back into making non-deoxy species by loading the patient with serine, the normal substrate for the complex, just driving it away from using alanine instead of serine, so to kind of correct the substrate shift. And that's a now established therapy for HSAN that's caused by variants in SBTLC1. We'll come back to that. But what was going on in our patients here? So this is what I just described to you in HSAN. We have a substrate shift from serine to using inappropriately alanine, and you get these toxic deoxy species. And when we looked at the biochemistry in our patients, what we found is something very peculiar. So here in the dots here are the HSAN variants, and in the diamonds are the ALS variants. And you can see here the deoxytoxic species are high in the HSAN variant, as expected, but normal in the ALS variant. But the canonical as complex fatty acids like ceramide and syngenine are high in the ALS and normal in the HSAN. So something completely different happening to the complex, and as opposed to having this abnormal deoxy species, we have much more of the normal canonical results of the pathway here, syngenines and ceramides that happen. And you can recapitulate that in cell culture as well, where you can, when you transfect the mutants for ALS, you get these high normal species, and when you transfect variants for HSAN, you get the high deoxy. So kind of a biochemical dichotomy that happened here. And that really led us to postulate that this is caused by excess sphingolipid synthesis in these patients. And that was also recapitulated in iPS-derived motor neurons by Matthew Niles and Payam Mohassel in the lab, where they could show that the motor neurons derived from ALS mutants have overproduction of these canonical species of sphingomine and ceramide. It's active in the motor neurons as well. So why is this happening? It's been long known that there is a feedback inhibition to the complex by the outcomes of the pathways, in particular sphingosine and ceramide, feedback inhibit the complex. It was Teresa Dunn across the street at Armed Forces Medical School who nailed down that this is dependent on one of the small regulatory subunits, the OMDL, and on the transmembrane domain of the complex. And she came up with this model where the OMDL, a small subunit, senses ceramide, reacts to it with dephosphorylation, and then blocks the SPT activity as a mediator of the negative feedback loop. And it does so by interacting with SPT in the transmembrane domain. And we could see the same in the cells from our patients. So here are cells from the patient, and we're trying to inhibit the complex with ceramide. In the wild type, you see the complex gets shut down at about 5 micromolar. But in the patient cell line, that shutting down is much dampened. Also, if you add the small inhibitory subunit, the OMDL, it only incompletely downregulates the complex. It seems to be no longer listening to the ceramide and the OMDL negative feedback. And when we showed our variants to Teresa, she immediately knew what was happening just looking at where they're located. All the ALS-causing variants are at the interface between the SPT complex in the transmembrane domain and the small regulatory subunit, the OMDL. So the negative feedback loop was interrupted, and the high ceramides and the high sphingosines could no longer inhibit the complex, which basically got stuck in overdrive. It didn't regulate anymore in response to what it was actually making. And it's very, very interesting how the biochemistry really follows the phenotype. This is Thorsten Hornemann in Zurich, and he does lipidomics here. If you look at the hereditary sensory neuropathy variants, the normal species are all kind of down, but the deoxy species are hot red, and the opposite in the ALS. And then he looked at patients who had been described previously as having both a motor syndrome and a neuropathy as kind of HSAN plus patients. But you can clearly see they have motor symptoms as well, always kind of referred in the literature as the 331 syndrome. And when you do the biochemistry on them, you can see they actually do both. They have high deoxy and high canonical species. So the biochemistry really follows the phenotype very, very closely. So you could ask, what about serine supplementation? Is that an avenue that we should be taking here? And you can already surmise, well, maybe that's not a good idea. If you have an overactive pathway already, if you go more of the natural substrate, maybe you drive it even more in overactivity, and that will be counterproductive. And that is exactly what Payam found. When he took his cellular models and fed it serine, the biochemical phenotype worsened. And that is highly relevant because some of the patients with these SPT-LC1 ALS variants had already been put on serine because SPT-LC1 you need to treat with serine when that actually is absolutely contraindicated. So here we have a therapeutic implication right away. But what can you do about it? And so this is proof of concept that this is actually a target for precision medicine as well. And this, again, is Payam's work. He reasoned that if you have a dominantly acting variant and heterozygote individuals who have only one copy of the SBTLC1 just do fine. Maybe the approach I should be taking is to inactivate this dominant variant, just take it out of the equation and see whether we can restore regulation of the complex. And so he did that by siRNA, looking at this transcript here is a mutant transcript. Here's the wild type, and this piece is missing in the mutant. So he makes an siRNA that recognizes the mutant allele, but is not binding to the wild type allele, selectively knocking down the wild type transcript and not knocking down the mutant transcript and not the wild type transcript. And he's able doing that to normalize the biochemical phenotype in the patient cells. If you just look at this first one here. So with the siRNA, the overproduction of the ceramide sphingosine species is shut off. So just taking out this non-regulated subunit normalizes the biochemistry of the complex as a proof of principle for a precision approach to this. So it also meant that we should revisit patients that we had seen with a juvenile ALS phenotype, such as this young boy here, who had an even more severe course of progressive weakness, denervation, upgrowing toes, you can see here, and even earlier onset of this juvenile ALS phenotype who had remained unexplained. And what we found in him is a de novo mutation in the other subunit of SBTLC2. And that, again, has expanded now in collaborative work that there are variants in SBTLC2, the second subunit, that also have this dichotomy of HSAN mutations and distinctly ALS variants as well. And the biochemistry is exactly the same for SBTLC2 as it is for SBTLC1, which is overproduction of the SPT canonical readouts products. And again, the answer for that is in the structure. So also the SBTLC2 variants interact with the ORMDL interaction with the complex. So the same phenomenon, but by a different subunit. So one kind of simplistic way to look at it is as a first therapeutic approach is, so maybe we can just downregulate the SPT complex overall in all of these variants that cause overactivity. And there are compounds like myriocin, which is a fungal compound that would be able to do that. And so we're studying that now to see whether this small molecule would be applicable as a therapy for this very severe phenotype. So for this first story, I'd like to acknowledge Payam Mohase, first of all, but also Sandra Donkervoort, our genetic counselor who was driving the discovery, Matthew Nile, Torsten Hornemann, Teresa Dunn, for sure, across the street, and many international collaborators that have worked on this. The second story moves us to muscle now. We're going one step further, and we're seeing a young man with something that we actually know and think we know, which is Ullrich congenital muscular dystrophy, and I will tell you what that is, but who remained mutation negative despite whole exome sequencing and next generation approaches. So this is this young man that we encountered together with his local team, and he has a story that he had not much to show in terms of symptoms at birth. He walked at almost the appropriate age, but then had rapidly progressive weakness as well as contractures and hyperlaxity in his joints, developed scoliosis and respiratory insufficiency that required non-invasive nocturnal ventilation. You can see here this very peculiar coexistence of striking hyperlaxity in the distal joints, but very contracted proximal joints. So he's really contracted in the shoulders and the elbows, but has very striking distal hyperlaxity there as well. He also has abnormal skin and abnormal scarring. Now, that's a phenotype that's so eminently recognizable that we immediately say, well, this is easy. This is this disease, Ullrich congenital muscular dystrophy, described by Otto Ullrich in the late 20s, early 30s in Munich. And he called it sclerotonic muscular dystrophy because of this coexistence of sclerosis, muscle tightness, and hyperlaxity. It is so strikingly unique that this is his original publication. This is a modern day patient. That this is we say in German, Blickdiagnose. This is, you walk into the room and you know what this is. That's as striking as that is. The only problem here was this young man had no mutation to show. He had whole exome sequencing. There was nothing to be seen. So we come back to that later. So what is the spectrum of collagen 6 muscular dystrophy? So it comes as a phenotype Ulrich that I just described to you, very early onset, quite severe. Infants may not walk, or they just walk on their knees because of the contractures in the knees, or they walk for brief periods of time and then lose ambulation because of ongoing weakness and the contractures that you see here. And there's a more intermediate phenotype where patients do walk with considerable weakness into adulthood but have early respiratory failure. You can see maybe that she has a tracheostomy because she has nighttime ventilatory failure. And then at the mildest end, we have this muscular dystrophy type Bethlem, where patients have the contractures but less weakness, and they walk into adulthood and have more stable respiratory situation. But they also have skin findings here, such as the keloid formation here. And these three phenotypes overlap, but they're clearly hallmarked by the degree of respiratory decline over the years. This is a big retrospective study that Regan Fody spearheaded here and at UCL in London. You can see the Ulrich patients have this steady and progressive decline in respiratory function. And if one doesn't intervene with non-invasive ventilatory support, they would all die in the teenage years. But with the ventilatory support, they do survive milder, longer. And then the intermediate and Bethlem phenotype is milder. So what's happening to muscle here? That's a normal muscle. You can see the muscle fibers neatly outlined, very little connective tissue in between the fibers, all the myonuclei at the periphery. And this is a biopsy from one of these patients. You can see this is like a destruction happened in the muscle. You have lots of connective tissue now encasing the muscle fibers. Nuclei have moved to the center as a sign of regeneration, and you have lots of fat replacement in the muscle as well. So we call that a muscular dystrophy because of the regeneration and degeneration going on at the same time. This type of muscular dystrophy, however, is special because it's not caused by a defect in the muscle. It's actually caused by a defect in the extracellular muscle, in the extracellular matrix that encases muscle. This is a transmission EM picture where the muscle fibers have been extracted, and this is the matrix that's left behind. So it's like a glove hugging a fiber that's left behind. And this is where collagen 6 lives, in this extracellular matrix, both surrounding every muscle fiber, but also in the epimysium, perimysium, and then also in the tendon connective tissue that connects to bone. Collagen 6 is this kind of beads on a string structure, molecule that lives in these matrices, hugging the muscle, interacting with the muscle immediate environment. And this immediate environment of the muscle fiber here that's shown in this diagram here is extremely important, not only for mechanical support and force transmission, but also for the regulation of growth factors that reach to muscle. And the deficiency of collagen 6 leads to both structural problems, but also to problems with growth factor regulations that then have a secondary effect on muscle, causing the degeneration. And fitting with that as being an extracellular matrix disease, collagen 6 is actually not made by muscle fibers in the muscle, but rather by the fibroblastic adipogenic progenitors, which are mesenchymal stem cell-like cells that are the matrix producers in muscle. That's where collagen 6 is made, and you can see it's made from 3 genes that form a monomer, a dimer, and a tetramer. It's a huge protein structure, as big as a human ribosome, and that gets secreted into the extracellular matrix to form this collagen 6 network. And you can see in single nuclear RNA-seq, for instance, that really it is these interstitial cells and tendon cells that make the collagen 6. Genetically, there's 2 mechanisms causing it. You can either have recessive variants that knock out collagen 6 production completely, so you're null, you have no collagen 6, and you have a severe phenotype typically of Ulrich. But much more common is this phenotype, the dominant negative mutations that cause an abnormal collagen 6 that gets secreted but no longer connects to the basement membrane. Basement membrane is labeled in green here, collagen 6 in red, and you can see normally they tightly overlap, but in the dominant mutation that interaction is disturbed. You have lots of dysfunctional collagen 6 sitting out there. And why is that happening? And that goes a little bit back to the assembly of collagen 6. So we have 3 collagen 6 genes that form 3 monomers, 3 chains that form a monomer, that form a dimer, form a tetramer, get secreted. In a dominant mutation in one of these chains, and if the dominant mutation is able to assemble the entire pathway, it really poisons the assembly successively, so that at the tetrameric state you only have 1 out of 16 tetramers that is wild type, and hence you have a severe collagen deficiency in the matrix. So these assembly competent variants in any of the chains causes severe dominant negative phenotype, and that's shown here schematically as well. Typical variants that we've known were in-frame exon skipping variants or glycine missense variants, always in the same spot in the triple helical domain that allows the chain to be assembled into the assembly process. That's key for the dominant negative process. It has to be assembled to cause this striking imbalance of wild type to mutant. So why am I telling you all of this? Because we have to come back to our patient here. So at the time, we were working with the broad to see, well, maybe with the advent of transcriptomics, maybe we can figure out these patients who look like they should have something we know, but we can't find mutations. And so we worked with the team at Broad, with Francesco Montoni in London, to see whether our patients could be solved using RNA-seq. And so RNA-seq from the readout looks a bit like this here in a control sample. All the reads that you find in RNA-seq are faithfully mapping back to the exons of the gene, just like you would expect. You know, the messenger RNA being processed and the sequences need to fit to the exons in the genome. But what we found in 4 biopsies of these patients where we were absolutely 100% sure of the phenotype, all 4 patients from 2 continents showed the same thing on the RNA-seq. All of a sudden, a piece of the intron that doesn't belong into the messenger RNA is showing up on the RNA-seq. And that was the first kind of shock, but it was shocking to see that all 4 patients actually had the same deep intronic variant, absolutely the same variant, de novo occurring in the deep intron that activates a cryptic splice event. So it creates a new splice donor and activates a cryptic splice acceptor here. All 4 patients had the same here. And it's now, and that was because it's so deeply intronic, was not seen on whole exome sequencing at all, where you only look at the exome sequencing. And not only that, it is now one of the most common collagen 6 mutations causing disease. Up to that point, completely undiscovered. And now we have a series that is pushing 50 patients, all with classic and severe Ulrich congenital muscular dystrophy here, put together by Regan Foley, that have this very consistent phenotype that is in Ulrich muscular dystrophy, but it starts a little later, but is more progressive, as if there's kind of dominant negative effect that builds up, kind of accelerates over time as a sub-phenotype of this. So back to what's happening here. So you have this variant that always is the same de novo variant that activates the pseudoexon from the intron to be spliced in. And when it gets spliced in, it is, when it's faithfully spliced in, it's an in-frame pseudoexon that's translated into protein. You get a little bit of splicing where the intron is retained, but that's out of frame and doesn't cause a dominant effect. And when you look at this just from a genetics point of view, a precision therapy is kind of delivered to you on a platter. And that precision therapy is, I'm going to get rid of this pseudoexon. I'm going to skip the pseudoexon. I'm going to splice modulate it using, for instance, ASOs that mask the splicing event, and that allows me to go back to wild type. So the ideal precision medicine target, because with this splice correction, you are actually creating wild-type transcript by suppressing the pseudoexon. And that's just kind of on the drawing board. If you just look at it, this is what I want to do as a kind of precision approach to this particular variant and tailor-made to it. And so that's what Veronique Bolduc has been driving in the lab. Here's the diagram again of the splice events. Again, the pathogenic one is the one here where the pseudoexon is in frame, and it actually gets translated in the triple helical domain, in the collagenous domain where you have this repeat glycine XY motif in a completely random peptide that interrupts the triple helical domain. And it's exactly in the spot where all the dominant variants are that we've previously known. So it's assembly competent. It gets assembled into collagen 6. You can make an antibody to this. It shows up in the matrix. It's made into protein. It massively messes up collagen 6, maybe even more so than the traditional dominant variants that we had seen. So an excellent, maybe difficult, but excellent target to address with splice modulation using ASO. And so Veronique has made several PMO ASOs that cover the pseudoexon sequence as well as the splice sites, was able that she could, with the ones that live actually that in the pseudoexon itself, probably suppressing splice enhancers in the pseudoexon sequence, she can suppress the pseudoexon inclusion that you see here showing up and revert back to the wild-type transcript. In cells, that is, of course. And you can, with that, suppress the peptide, so the translated pseudoexon in culture, and you can normalize the extracellular collagen 6 matrix that is made by these patient-derived cells here, much less interrupted, much more straight. So we have a target, we have an active compound, and now, of course, we have to get back into patients, and not only that, but we have to get back into these fibroblasts where the collagen 6 is made, which is the next kind of challenge that we have to address with it. Are we going to move the goalpost with that for the disease? Well, again, a clinical observation tells us, yes, we might, because we saw this patient here who is much milder, you can see on muscle MRI here, compared to a normal, to a fully expressed collagen 6 pseudoexon patient. All the white is fat replacement, and so her muscle MRI looks more preserved, her weakness is less, her phenotype overall is less. And what turns out to be the case is she is a somatic mosaic for this deep intronic mutation. So she has two populations of fibroblasts, normal ones and diseased ones. And if you look at that kind of in nature suppression of the pseudoexon by state of the mosaicism, you can see it actually shifts the phenotype towards milder. That gives us hope that suppressing the pseudoexon variant in patients will be therapeutically relevant. And that's work from Regan, Ying, and Riley in the lab. Meanwhile, Veronique Barrett and Fadi in the lab have gone ahead and made a mouse model for this variant. This is intronic, so you can't rely on mouse introns for that. You have to replace the entire region with human sequence, engineer that deep intronic mutation in there, and luckily and faithfully it recreates the pseudoexon inclusion in the mouse as well. Mouse has a phenotype, it's weaker, and so now we can use the mouse to target our therapies here as preclinical testing, but we also can test other approaches to the pseudoexon, such as cutting it out with CRISPR-Cas, just cutting out that piece of intron that's not otherwise needed and get rid of this mutation. And so that's going forward now. So for this story, really the drivers were Veronique in the lab and Regan in the clinical, Ram with lots of other people helping us along, in particular Sandra again, Ying, Fadi, who's working on this very much. In collaboration with the Broad Institute, collagen 6 specialist in Cologne, Raimund Wagner, the UCL group under Francesco Montoni, and then many international clinical collaborators that contributed to the series. So the final leg of our journey through the neurogenetics of motion and sensation brings us now to the sensory arm of this loop, and that is, again, originating from one single patient, a young woman that we saw in Calgary, in Canada, in an undiagnosed disease clinic. It's Mike Innes and Jean Ma, there for either a suspected neuromuscular disease or a movement disorder. She was kind of seen in both clinics for movement problems as well as for neuromuscular problems. And her story is quite unique. So she was diagnosed early and as having low muscle tone. As a newborn infant, she had difficulties starting to breathe. So she had respiratory adaptation problems. She needed intermittent ventilatory support, but that then kind of normalized. But she had severe delayed walking. It took her 7 years to start to walk independently here. She had early problems with fine motor skills. She had difficulties manipulating buttons and developed early and very progressive scoliosis during preteen years already, and finally needed surgery at 17. So she had, on examination, she had a normal cognition, was very smart, is now in college. And this is why she ended up in a movement disorder clinic, because she was not able to hold her hands still. She had what we call arthritosis, or better pseudoarthritosis, as you'll see, where she just couldn't control her movement. They had this constant writhing movement in her limbs. When she was walking, when she had a certain stride velocity and stride length, she was actually quite efficient at doing that. But when you put her in the gait lab and close her eyes to take away vision, and you have to put her in a harness, in a moment you see why, is because then she loses all sense of walking and collapses to the ground and is no longer able to even hold herself upright. And that's a young patient that we saw just shortly after that, who is not even at the walking stage yet, with the same kind of problems keeping herself upright when the eyes are closed. So, formal neurological examination, she had a strikingly positive Romberg test that you just saw in this video, but she also had absent proprioception and testing of joint position. And not only in the distal joints, like we often see in a neuropathy, in any joint. So the shoulder joint, the hip joints, any joint you move, she had no sense, absolutely no sense where it was in space. As if she was completely deafferented, no feedback from the joints, and that's why you see these pseudoarthrotic movements, which are really deafferentiation because she has no joint position. She also had poor fine motor control and absent vibratory sense, and she also has a sense deficiency in touch, and that I will show to you in a second, but that wasn't really clear to me when I first examined her. So in her case, whole exome sequencing landed on this gene here, which is piezo2. And that's a channel. It's the largest channel in the human genome. You can see it has this enormous propeller-shaped leaflets that kind of embed in the membrane and control a central core with a cap on top, pore function with a cap on top of it. And what it does, it immediately senses any small deformation in the matrix, in the plasma membrane mobility and deformation, and reacts with an opening of the pore and a calcium flux. So it's a mechanosensor of the membrane. It's very, very sensitive to smallest deformations of the plasma membrane, reacting with an ion flux to it. It is particularly prevalent in nerve endings in the skin, such as the myoclonic but also in muscle spindles, where proprioception is picked up in the muscle. These fibers are all highly expressing piezo2. So when we got this genetic result, we weren't quite sure what to do with it. And then I was in a faculty meeting, and Jeff will excuse me, but typically you fall asleep in a faculty meeting. You hope it's going to be over very soon. But the core facility, the microscopic core facility, showed the new confocal that they had acquired, and there were beautiful images of a dorsal root ganglion showing piezo2. And I said, well, who made these images? And it was Alex Chesler, who was a Stratman investigator who had just joined the NIH and turns out to be the expert in piezo2. And I emailed him, can you help us with these variants? Can you help us to understand this? And he's in building 35, one floor down, and he dashed up the stairs, and we sat in the office, and he really explained to me what might be going on here. And so we were able to bring these first two patients to the NIH to really deeply phenotype them with that in mind. to really describe for the first time what it looks like if you have no piezo2 in a human and no proprioception at all. So you can see here on this quantitative testing for joint position, the patients in red are completely random. Their choices are 50% right, 50% wrong. If you do that on yourself, and I encourage you to do that, you'll be never off. Even with the smallest movements of distal joints, you are absolutely 100% accurate doing that. What the patients also had, which I hadn't realized, is they had a touch discriminatory defect. So they were unable to do two-point discrimination in the glabrous, kind of the non-hairy skin, which you might call the analytic skin. That's how you touch your environment and feel objects. And they couldn't feel an object placed into the hand, so didn't know whether it's a cube or ball or key. And they actually then said, well, yes, now I understand. This was actually always the case. But they didn't have this defect on the emotional skin, which is the non-glabrous skin. So when you try to comfort someone, you stroke the hairy skin, you stroke the head, and that's a very different neurobiological skin from the analytic skin. And this defect only was in the glabrous skin. As opposed to that, completely normal pain sensation, temperature thresholds, and you'll see more of that a little later. So very selective sensory mechanosensory deficit that these patients show. And the clinical consequences are mostly driven by the proprioceptive defect. That causes a severely delayed ambulation, the early scoliosis, the joint deformities, and the coordination. So what is proprioception? So it's kind of popularly known as the sixth sense of the human body, the kind of hidden sense of the human body. But it's a very interesting sense because you can consciously experience it. If you close your eyes and you move your hands and you touch your nose, you can consciously follow what you're doing and where your limb is in space. But you can never turn it off. So you can't close your eyes. You can close your eyes for vision, plug your ears for hearing. You cannot turn off proprioception. It's always on. It's prenatally on. It is always on to keep your spine upright in space. It is a sense that you absolutely rely on for body coordination, but also body alignment. And when we look back at the little infants born with this piezo2 deficiency, they already had signs and symptoms. They had dislocated hips, joint deformities at birth. So prenatally, it already kicks in. And without proprioception, you would never be able to do this, or gymnastics and dancing. We've now assembled a much larger cohort, and with a very, very consistent phenotype. And the most striking part of the phenotype is that all patients had this early respiratory adjustment problem. Come back to that. So as if there was a mechanosense deficit in the lung inflation as an infant as well. They all have early and rapidly progressive scoliosis, even though the muscles look normal, and they had these progressive joint deformities as well, even though there was no muscle weakness involved. To give you a flavor of the phenotype, just show you again what it looks like. With eyes open, you can see this young man walks by fixating his eyes on the feet. So he knows exactly where he places his feet over time. This is a different boy. When you close the eyes, he will immediately sway and then collapse to the ground, being caught by the harness here. You can see the pseudoathetosis again, as edema sade, with me there with this patient. And he's closing his eyes, and we ask him to sit absolutely still. He cannot. So his joints and muscles are moving constantly because he's lacking any feedback for it. This is a video a gentleman sent us from Holland to show to us to see whether we think he might have this. So he's touching his nose with his eyes open, and now he's closing his eyes and see where he goes. He has no sense whatsoever where his limbs are in space. So Alex has actually made a mouse model of this with selective knockout of piezo2 in the sensory neurons. And you can see here this mouse has no clue where the hind limbs are, doesn't know where to place them. It's kind of just sitting there frozen because there's no movement possible, because there's no visual control of the limbs for this mouse. And you see they just go off in space because completely deafferented. It is strikingly similar what we see in humans as well. So this is a 6-year-old who still can't walk, and she can't even crawl properly because she doesn't know where to move and where to place her hind limbs, her legs in that situation. It's very interesting. So patients have told us, a very eloquent patient from France said, she said, when I close my eyes, I lose my body. I think this is really very evocative of what's happening here. And another 2-year-old, the mom reports, when she wakes up the 2-year-old in the morning, pulls away the blanket, the little one looks at her feet and says, oh, hello, feet, just after visual control. So I've been thinking, what can we do about this? And because it's such a selective sensory deficit, we figured maybe one thing to do is to try to circumvent the proprioceptive defect by using a different sensory modality that the patients actually can experience. So here, schematically, in a normal case, you can move your water glass to the mouth with your eyes closed because you have proprioceptive feedback. In a patient with piezo2 deficiency, you have to stare at the glass and control the movements with visual control at all times. And the idea would be to have a goniometer that measures the joint angle and velocity and translates that into a sense the patients can actually perceive, which is deep pressure and temperature, and then they can learn to remap proprioception to the sensory input that is preserved. And so we're working on that at the moment with Tom Boulia in the Gate Lab here and Alison Okamura at Stanford in the Charm Lab to start with a very kind of clunky first-generation device for the elbow where we can test the reaching test like we do here to see whether we can correct this. A little patient from France, we told her this and that we wanted to have this for the legs eventually to allow patients to walk earlier. She's from France. She said, well, these are pantalons magiques, and she drew them out for every age from 7 years on to 4 years on to 60 and completely grasped the concept of it at 6 years of age to allow patients like this one to then be able to perceive where the legs are and learn walking earlier than she did. So in the last little bit, if you bear with me for a few minutes, I want to bring this back to the lab. So Alec Nichols, when he was a graduate student in my lab, decided to model this in cells and to devise a way to create sensory neurons out of skin cells. So he came up with a protocol of programming of these iPS cells to drive them towards a sensory neuron phenotype. And what he found is that if he had short programming, he can create a pure touch receptor. If he had long reprogramming, this touch receptor also perceives cold. So he predicted that these neurons might exist. And now we know they actually exist in the trigeminal system. We have neurons that perceive both touch and temperature in the face in particular, which are probably ancient sensations to know. They know the cold wind is touching your face, and that may be an important sensory input that we had evolutionary. So the way he measures that is by, so this is what these look like, the nice DRG-like looking cell, and you indent it with a pipette, and you get this immediate, fast, and rapidly recovering current. And in the patients, this was absolutely flat. They had no mechanosensation. You could poke them, and there was no current. If you CRISPR correct the mutation back to wild type, the normal current is reestablished, really showing that in the patient-derived cells, it's purely and only dependent on this piezo2 deficiency. And so he can identify these patient-derived induced sensory neurons as completely insensitive to mechanical stimuli. And they are pure sensory neurons, so they perceive coldness just fine. So I told you about mechanosensation. I told you about touch. I told you a little bit about interoception mechanosenses as well in the little infants that can't inflate their lungs at birth, probably because the Herring-Breuer reflex is interrupted. That also is mechanosensitive. But the phenotype keeps expanding, inspired really by mouse work and then translated back to humans, where every single thing that was found in the mouse is actually replicated faithfully. So that was a really astounding collaboration with Alex Chesler and Ardem Pataputian. Ardem Pataputian got the Nobel Prize for discovering this channel, and it's been a very, very productive collaboration where resulted in this string of papers in collaboration with the clinical phenotype, really, to map back what was seen in the mouse to the human. Just to give you two flavors of that, this is a pain sensation that actually is absent in piezo-2 patient. That's allodynia. Allodynia is the pain you feel on burnt skin. So if you have a sunburn and you touch that skin, it's painful. If you burn your skin, you touch it, it's very painful. A huge problem in burn victims. And the question is, what about this pain? Because it's a touch pain. What happened in piezo-2 patients? And so that is in this paper here. And so the test for that is very simple. You induce mild inflammation on the skin with capsaicin, and then you touch a control area of the skin and the inflamed skin and ask the individual whether this feels painful. So at baseline and inflamed, so in the normal individuals, they're 100% correct. They always correctly identify the inflamed skin. But the piezo-2 deficient individuals are random. They don't feel this as painful at all, meaning that allodynia is dependent on that functional touch sensor. Why is this causing pain? So I'm also play electric bass, and so in electric bass you have a pickup on the bass and you have an amplifier, and you can do the same plug with the normal pickup and create a nice sound. If you turn up the amplifier, it gets a screaming distortion. And I think the same is happening here. Piezo-2 is the pickup, perceive the touch, but then a pain loop gets into overdrive and causes this excruciating pain. It's not caused by piezo-2, but by the summation of inputs in the central loop of pain sensation. And then also in urination, so Ardem had found that if you knock out piezo-2 in the bladder, the mice don't perceive bladder fullness anymore. And we went back to our patients who had never volunteered this, but when you actually asked them, they said, oh yes, I always put my clock so that I go to certain times so that I don't have overflow. Because they've lived with this all their life, they've never perceived it differently, and so they've never volunteered us to us, and we never asked. But when you specifically ask, this comes up as a phenotype as well. And this was again done by Dima Sade. And this is just a homage to these patients and their resilience. This is a Special Olympics athlete with piezo-2 deficiency who designed his wheelchair that he can power it without needing proprioception. You just can't stop him. So I think I've tried to convince you that every patient has an important story to tell and can guide you to new and unexpected insights. But you have to listen to the patient and examine very carefully. And I was really fortunate in my training to be exposed to, I think, two giants of classical neurology at MGH who were still active when I was there, Ray Adams and Miller Fisher, who are really unbelievable clinical experts in identifying any subtlety of the neurological syndrome in front of them. These are clearly pre-genomic neurologists, very clearly so, but the message and the lesson, the skill set that you get from clinical giants like that will not expire. And I would propose that even in our next generation genomics era, clinical skills remain critically important and may even be even more important to keep this wheel of phenotyping and genotyping honest and moving and arrive at precision therapies. I should also mention that whatever starts local is immediately global in our field. These are two other kind of discovery stories that I won't belabor, but it tells you when after seeing the first patient and broadcasting it into a network of collaborators and friends and experts, you immediately have a global team to work on that. On the flip side, I've also been globally active in many, many clinics for patients with unclear neurogenetic diseases, and it becomes very clear that there also is a global imbalance to access to testing, to access to therapies, and even as simple therapies as respiratory care. So even though we are a global team of geneticists, the inequality, I think, globally remains striking and needs to be addressed. For that, we have a network of superb clinical colleagues. So I'd like to, again, thank the entire team. This is just the more recent team, and I acknowledged people as I went along that really made this happen, as well as our NINDS leadership team, Story Landis Walter, who knew me as a resident, and despite knowing me, got me to NIH. That was very nice. Nina, and then the clinical director, Avinat, and scientific directors, Ellen Lorna, and now Jeff Diamond. And a big shout out to our team as well. This is how we started at CHOP, Children's Hospital of Philadelphia. And Yachin and Ying are still here and with me in our current state. So my journey is really going south. I started at Boston Children's and MGH, then went on to Children's Hospital of Philadelphia, and then to NIH here in Washington, D.C., 15 absolutely formative years that really got me on this trajectory. And now moving on next year to St. Jude Children's Research Hospital to chair a new department of genomic and translational neuroscience that will take this concept of individualized diagnosis and therapeutic concept further and trying to make impact on even the rarest of patients. I thank you for your attention.
Well, there will be a reception, so I'm afraid we'll have to hold the questions for the reception. Sure. But one question for you online. It's a quick one. How important is oxidative stress in these motor neuron diseases?
That's a really good question. So probably is very important. Where Payam is trying to figure out at the moment what actually happens if you have this sphingolipid overload. And so one thing that probably went through all of your minds, and I have no answer for, is what is the selective vulnerability? Why are sensory neurons vulnerable to the deoxy and deoxymethyl species, but not motor neurons? And why motor neurons so sensitive to the overproduction of the normal sphingosine and ceramide and downstream components of it? We don't know what that vulnerability is, but there is emerging evidence that it has something to do with mitochondrial membranes, and those are, of course, exquisitely vulnerable to oxidative stress as well. And so there could be a summation effect that will then happen.
So there's a reception right outside.
Before the reception.
Oh, yeah, please.
Wow, what an insightful lecture, Dr. Bonnemann. I know you told us at the beginning that you were going to describe your 3 personal journeys. From really an enigma encountering patient to actually discovering new approaches and new treatment to treat neurogenetics and neuromuscular disorders and diseases. On behalf of the Clinical Center and the NIH writ large, I want to say thank you so much. And please join me in giving him a very warm round of applause.
Thank you.